Stanford University.
So with that, it's a great pleasure for me to introduce today's speaker, Raj Kishore. He's an outstanding scientist, he's also a good friend of mine. He's going to talk about stem cell exosomes and myocardial repair. By way of background, Raj obtained his bachelor's degree as well as a PhD degree at University of Lucknow in India, and then he came here for his postdoc. He was initially trained as a immunologist. There's more sheets in the front. Initially trained as an immunologist, working on microRNAs for inflammatory gene expression, and also working on tumor antigen presentation and immunological memory. So he did 2 postdocs, one at the New York Medical College, and then another one at Cleveland Clinic Foundation. And then afterwards, he essentially transitioned into cardiovascular research. He was hired by Doug Lasordo, who was probably a speaker here maybe about 2 years ago, to be instructor in medicine at Tufts University. And then he was assistant professor there before he moved to Northwestern University from 2007 to 2010. And then just past a couple years ago in 2014, he relocated to Temporal University to be the director of the stem cell program at the Center for Translational Medicine. I've known Raj for a long period of time. You'll know that he's passionate about training the next generation of physician scientists. I counted on his CV, more than 10 of his trainees have become faculty members throughout the country and throughout the globe. He also has more than 100 papers, many of which in top impact factor journals, Nature Medicine, PNAS, JCI, Circ, and Circ Research. He's been continuously funded for the past 15 years, including currently 3 R01 grants. He also has 7 patents related to microRNA, exosomes, and also immunological research. And I would say he's one of the leading experts in this area for exosome research. If you Google exosome stem cells, his name always pop up. And from a personal standpoint, I just had the pleasure of knowing him, interacting with him for the past 7 or 8 years. I always learn quite a bit of stuff from him every time when I talk to him. And it's a great honor for us to have him here and give this talk today. Thank you, Raj. Yeah.
Can you hear me? Works? Works? Thank you, Joe. Thank you for the invitation. It's my first visit to Stanford, and it's always nice to see old friends and make new friends. And so I got to know Joe. Joe was the person who killed my first R01. I still remember that. I got to know much later, but it's all good now, Joe. So it's all right. All right, so I would like to begin with the most important slide of my talk, and that is to really acknowledge upfront the people who do the work. It's not me who does the work, but these are the guys who work really hard in the lab, and this is what I end up doing. I'm the beneficiary of the hard work, so I just want to put this thing first and acknowledge the real hard work of the people who do all the work. All right, so in my lab over the last 10 to 14 years, what we are working on the different aspects of the stem cell function. One of the areas that we are working actively is to use the anti-inflammatory cytokine interleukin-10 to really inhibit the inflammation, do a co-therapy with the stem cells to enhance their survival and function. And we have published a number of papers on this aspect. We are also working on epigenetic modification of the little stem cells to enhance their plasticity and function. And we have used both small molecules and the genetic method, and we published on that area too. We have also delved into nanobiomaterial-based enhancement of retention of the stem cell that transplanted after the injury. And last couple of years, or 3-4 years, we've been working on the exosomes as a cell-free modality for the augmentation of endogenous myocardial repair, or replacing the cell by using the exosomes. So today I'm going to talk to you about the exosome biology and some of the published work and some of the unpublished work that I'm going to show to you. So I'll tell you some background about exosomes. I'll tell a story that's published about embryonic stem cell exosomes, immediate myocardial repair, and some ongoing work after this publication. And then I'm also going to tell you that we have to be careful that before we take the exosomes to clinical trial, we have to really understand whether all the exosomes are created equal or they might have a detrimental effect too. So it's the cautious tale. So the question that all these cardiac degenerative people have been asking for many years, can the heart repair itself after the injury? And the short answer is it tries. It tries to help itself, but it's very inadequate. What's the proof? We know that after the injury, there is a low replication of the myocardial proliferation, but these are terminal differentiated cells, and the proliferation capacity, if any, is way too low to make any physiological meaning. We also know there are resident cardiac stem cells in the heart, which differentiate to different kind of cardiovascular cells. But these cells have inadequate proliferation and differentiation ability. They're less plastic, and there's less chemotaxis of these cells to really home to the area of injury, and so they're also inefficient without being manipulated. We also know that bone marrow stem cells mobilize to the injury where they do vasculogenesis, but the mobilization is also defective in response to the inefficient homing of these cells from bone marrow to the heart. And of course, they are also unipotent and not the multipotent cells. So heart tries to repair itself, but all this process combined are inefficient to really repair the heart after the massive myocardial infarction, the loss of billions of cells. So given the high mortality, we know the endogenous mechanisms are very inefficient, and that led to the transfer of the exogenous stem cells, mostly adult stem cells, into patients, first in the clinical trials. A number of stem cells have been used in the clinical trial. People have used the different kind of stem cells, which have shown to activate the endogenous stem cells, differentiate to different cell types, inhibit apoptosis, reduce fibrosis, and improve the heart function. Till date, and this is an old slide, there's much more cells, a couple of more kind of cells right now. These kind of stem cells, including from skeletal myoblast earlier to the cardiac-derived stem cells and adipose stem cells and hematopoietic stem cells, have all been used in the clinical trial. And while we acknowledge that the results are encouraging, but the expectations were not met by the clinical trials in the adult stem cell therapy. And most optimistic people will say that they're encouraging, and pessimistic people will say that they didn't work at all. So stem cells therapy experience has not been as robust as we have seen in the animal studies. There are a number of reasons why adult stem cell therapy didn't work. First, we all agree that once you transplant these cells into the ischemic myocardium, there's an extremely low level of cell survival and retention. You'll be lucky to find 2–3% of cells a couple of days after you transplant them into the myocardium. We also know that adult stem cells have limited plasticity. They do not differentiate into all the cell types that's required for the healing of the heart. Specifically, their differentiation to cardiomyocytes is highly debated. What we have not appreciated until now, but we know that the stem cells themselves are dysfunctional because the myocardial disease is not a single disease, but age and the presence of the other metabolic diseases, such like diabetes and age, makes the stem cells themselves dysfunctional. And also there's a great deal of debate that how this adult stem cells work. Is it really a cell replacement, or is mostly a paracrine-mediated inhibition of the further cell death? So what we begin to think about was a few years back that can we use a cell-free system? Because as I mentioned, the stem cell, when you transplant into the heart, they do not survive more than 2–3%. So can we augment the more functional improvement by using something that cells might secrete and recapitulate some of the functional properties of the stem cells themselves in a cell-free system? This is just to show that a number of stem cells, the exosomes, have been used. Oh, what did I do? And the hope is that by the cargo, these exosomes might give the stem cell specific materials to the recipient cells and increase the proliferation of the endogenous stem cells, or reduce the inflammatory response, or they induce the myocyte proliferation and survival, as well as do angiogenesis. What are the exosomes? As you know, they are the small microvesicles that are secreted by most kind of cells, stem cells included. They typically range in the 30 to 150 nanometer size. And their biogenesis is not very well understood. But what is known is that basically that they are formed by the invagination of the plasma membrane. And then they assemble into the early endosome where the loading of the cargo, cell-specific material happens. They form into the interluminal vesicles where more loading happens and they become some multivesicular bodies. And then they have two fates. They're either very go to lysosome by degradation or they're secreted as exosomes into the interstitial surface. They have certain characteristics. All the exosomes do have certain cell surface markers, including CD63. They have some degree of MSC antigen as well, and they have some tetraspanins that help them to be fused to the plasma membrane of the cells and the endocytosis. In terms of the content, we get to know new things every day, but they're mostly loaded, very highly packed with the microRNAs coming from the cell type, as well as certain proteins and small mRNAs and other kind of RNAs. The way they work is the most common way that has been described in literature is by the fusion of themselves with the cell membrane of the recipient cells by endocytosis, go into the cell and release their cargo like a Trojan horse form of way. And these biomaterials that transferred from the exosomes, they mediate the downstream signaling component of the exosomal cargo. But they're also known to work by the juxtakine signaling mechanism, where they work in the classical receptor ligand pathway, depending on what cell type they're coming from, where the receptor and ligand classical signal transduction pathways. The importance of exosomes has really exploded before 2012. If you look at the PubMed, there were about 200 to 300 publications, but since the last 4 or 5 years, there are about 6,000 publications on exosomes in all the different fields. So just suggesting that the field of exosome biology has just exploded in the last few years. But if you do cardiovascular and repair and exosome, we are still lagging behind in terms of the exosomal research, with 20 publications or so, and most of them are review articles except some science articles. So a lot needs to be done in terms of cardiovascular application of exosomes. So the story began in 2011, where a young person, Sahu, in the lab and begin to work with the exosomes. And these are the exosomes from human hematopoietic stem cells, CD34 positive stem cells. And the idea was to see whether the exosomes secreted from these cells work as well as the cell. There's a couple of experiments shown in here where either the cell themselves or the exosomes derived from the CD34 human hematopoietic stem cells were used in an in vivo angiogenesis model, and this is a metrigial plug model in the mouse. And what you can see here is that the exosomes induced a larger angiogenic response in the metrigial plug than the cell themselves. In a second model, where Susmita used either the exosomes from the CD34 cells or a fraction of cells that were excluded out of CD34, just the CD34 negative fraction, and she showed. And this is a mouse corneal neoangiogenesis model system. And she went on to show that it is specific CD34 exosomes that drive the angiogenic response and not the fraction that's devoid of CD34 population. Many more papers came out since this first paper in 2011. And Eduardo Marmon's group showed that if you, there's a lot of data in this paper, but pay attention to this thing. If you stop the biogenesis of exosomes from the cardiac-derived stem cells that he uses by treating the cells with the small molecules that inhibit the ceramide system that's important for biogenesis, his stem cells no more works. So together, our data and data from other labs shows that exosomes are as potent in terms of driving the biological function as the cells themselves. But this data shows that the exosomes are the functional entity of a stem cell function in this model of myocardial infarction. Since then, number of other almost all kind of stem cells, exosomes have been described using mesenchymal stem cells, as well as bone marrow stem cells and c-KIT positive stem cells. There are certain advantages of using stem cells in the translational aspect. There's a strong evidence that exosomes are the main functional paracrine entity of the stem cells. We already know that they are cell-free nanocomponent and they mimic the stem cell therapeutic effects. Unlike the cells, these exosomes are very, very stable over the long periods of storage, and they are quite resistant to the changes in physiochemical microenvironment, including pH. If you freeze thaw them, they are much more stable and solid than the cells themselves. We also know they are very quickly taken up by the host cells, so they overcome the problem of stem cell survival, engraftment, and differentiation. And they can also be developed as a delivery vehicle of the desired cargo of small RNAs and protein of interest directly by manipulating the cell themselves or by manipulating the exosomes themselves. So a few years back, we begin to look at the embryonic stem cell exosome as a source of myocardial repair. And why embryonic stem cells? As I mentioned, the exosomes carry the functional cargo of the cell they derive from. And as we all know, that embryonic stem cells and the pluripotent induced pluripotent cells are really true stem cells. They have certain characteristics that are unique to them, not shared by the real stem cells. First thing is they have immense regenerative abilities. And so we guess that they might have very unique signature of cargo that can enhance the endogenous regeneration in the myocardium. And also the embryonic stem cells and iPS cells, they have a very unique cell cycle regulatory cargo, including the microRNAs that may induce cardiomyocyte proliferation and protection. Those were the hope to begin to work with this kind of exosomes. In the literature at that time, a couple of papers were published on the ES microvesicles. They were not called exosome then, which showed that the ES-derived microvesicle, they can reprogram the hematopoietic progenitor cells. And there was evidence to show that these macrovesicles can transfer the mRNA contained in them to the cells. And there was also another paper in some journal which showed that these exosomes or macrovesicles, they transfer the microRNAs from embryonic stem cells to the recipient cells. That's all was known by this time. There was nothing known for their repair abilities. So we begin by characterizing the exosomes, as we all do. The ES cell, this is mouse ES cells. Media was collected and exosomes were isolated by standard methods. We characterized, they are about 40 nanometer in size. And they were, as a control, we also used the embryonic fibroblast exosomes for a control, which is larger in size. They contain the exosomal protein specifically, so they are exosomes. And then basically they can transfer the pluripotency marker into the recipient fibroblast cell, suggesting that they are indeed coming from the embryonic stem cell. Then we tested these exosomes in the functional in vitro assays. This is just a cell death assay of C9 and C2 cells, where you put these cells into the hydrogen peroxide stress, and you treat them either with the ES cell exosomes or the control exosomes. And we're looking for, and these exosomes are now labeled with a dye to visualize them. We're looking for the activation of caspase-3 as a cell death. And what you can appreciate here is that ES cell exosomes specifically reduce the apoptotic cells and the activation of caspase-3. Also, if you put these cells on the endothelial cells and do a simple in vitro endogenetic assay, you can see this ES cells strongly enhance the tubulogenesis in the HUVEC cells when compared to the MEF exosomes or the media itself, suggesting that these are functional. And these exosomes do not form teratomas. So this is just a cell control where you put the ES cells in the minute mouse, they form teratomas. But up to 8 weeks of ES exosome transplantation in the mouse, both in the flank and the heart, we don't see any, not expected, we don't see any formation of teratomas, so they're safer, both in the heart and in the flanks. Then we use these exosomes to create in the LED ligation model of myocardial infarction and direct intermyocardial transplantation of these exosomes. As you can appreciate that, if you look at all the parameters of myocardial function, this is up to day 28, they were appreciably improved only in the mice that received ES exosomes and not the one that received the fibroblast exosomes. So there's a functional distinction between the stem cell exosomes compared to the controlled MEF exosomes. If you look at the structural evidence. The physiology was supported very well by the structure. You see the fibrotic area is decreased as well as the percent infarct size. And this is the picture of mesenchyme that's showing that the wall thickness is much improved and the fibrotic area is much decreased. This is a single injection of the exosome into myocardial, about 30 microgram protein equivalent of the exosomes. This looks all good. If you look further in the border zone, the animals that guard the ES exosome show a robust increase in the capillary density. These are all the small capillaries around the border zone, suggesting that there's a better perfusion when these exosomes are injected into myocardium. And you also see the great reduction in the depth of the myocytes in the myocardium. Now, what are these exosomes doing? I told you this, one of the characteristics of the ES and the iPS cell is the continuous renewal, very unique cell cycle with a short G0 phase. We did find the evidence of myocyte proliferation, and this is just a simple BRDU incorporation injected before the sacrifice in comparison to MEF exosome. The ES exosome induced the cycle activity marked here by the S phase entrance of the myocytes. Not only that, we also confirmed it by the phosphohistone staining, and this also showed the same results. There's a drastic about 3 to 4-fold increase in the cycling myocytes. Not only the myocytes, we also found the evidence that the ES exosomes very largely increase the number of resident cardiac stem cells. If you count the c-KIT positive stem cell in the two hearts, the ES exosomes recipient hearts have at least 3 to 5-fold increase in the number of c-KIT positive cells in the heart. And not only the number is increased, but the c-KIT cells are continuously proliferating. This is shown here by the percentage of c-KIT phosphohistone positive cells, as well as shown by the BRD as it goes up, that these c-KIT positive cells are expressing c-KIT, and they're also positive for BRD as a larger number than compared to the one that got control. So the ES exosomes are, in addition to providing the protection, they are working by inducing the cell cycle activity both into the cardiomyocytes as well as the resident cardiac stem cells. We were asked whether these c-KIT cells are coming from bone marrow, so we also stained them from the GATA4, suggesting that these are the resident cardiac c-KIT cells, and they are also proliferating continuously in the myocardium. So to summarize this part, what we have told you is that ES exosomes have the ability to induce survival and angiogenesis, and they also augment the cardiac function after myocardial infarction by largely inducing the cell cycle activity of myocyte proliferation and the augmentation in the number and proliferation of the resident cardiac stem cells. Then, because we saw a whole lot of augmentation of c-KIT cells, next series of experiments, we thought how these ES exosomes are prolonging or augmenting the function of the CPCs themselves. So we got this c-KIT positive CP cells from Mark Sussman's lab in San Diego, and this some in vitro experiment. This experiment just shows that if you take exosomes from the ES cell and put on CPCs, within 6 hours, you can see that they are all scattered in cytoplasm. By 18 hours, this whole cytoplasm is decorated by the exosomes from the ES cells. And then we did some in vitro assays. Shown here is that if you prime these CPCs with the ES exosomes, they tend to have a protective in the response to the stress of hydrogen peroxide. They have a larger proliferation, and they can also show the greater cardiac commitment, shown here by the RT-PCR of the cardiac specific genes when they are put into the differentiation media, and they also have the angiogenesis activity as I shown you before. So it's a function of the c-KIT cells, both in terms of their survival, proliferation, and their differentiation capabilities, is enhanced when they are primed with the ES exosome compared to the MF exosomes. Then we also prime these c-KIT cells, and we begin to ask this question that if you already pre-treat these CPCs and put them back into the heart, what happens? So this is just the experiment shown in here. So the CPCs were primed either with MEF or the exosomes, and they were transplanted back into the LAD ligation mall system. And this is just the functional data of the 8 weeks, showing that the CPCs themselves showed a trend towards better function, but not really significant in our hands. But once you prime these CPCs with ES exosomes, you can see there's a great separation and a much significant reduction betterment in the ejection fraction and fraction shortening, and is shown here by the structure. You don't see it's very large in FOC in these animals. It doesn't get better with the CPC alone, but if you prime them, you can see great reduction in the FOC size. It's quantified here. We also saw that the cells that have been primed by ES exosomes, they have a larger retention in the myocardium for a longer period of time, as well as proliferation, as I showed you before. So these cells which received ES exosome before they were transplanted are still cycling on the day 7 or 5 of the transplantations. You can find the cells in a large number, and they are still proliferating in the myocardium. And that's just the quantification of GFP phosphorhistone positive cells. It appears that some of these cells can differentiate to smaller myocytes in the myocardium. Their differentiation activity is also improved. I don't know whether they're bona fide functional myocardium, but they definitely express the protein markers of the GFP and the cardiac troponin, and they appear to be smaller in size. So it looks like they're trying to become myocytes, but whether it makes the functional difference by differentiation of these cells is not clear. But what's clear was that these cells definitely die less if you prime them before the exosomes. You can count them for a long period of time, and they definitely do differentiate to the vasculature. That's a much larger number in terms of SM22 positive arterioles compared to the one that were not primed with these ES exosomes. Then we begin to look at the content of these exosomes, and so like everybody does, we focus on the microRNAs. And we did the microRNA analysis of both the fibroblast-derived exosomes as well as the ES-derived exosomes. And what we found is, as expected, very different profiles. And these ES exosomes are enriched in 290 cluster of microRNAs, 290 family. And these microRNAs are pretty well known as the ES cell-specific cell cycle regulating microRNAs. And they contribute to the unique cell cycle of ES cell by directly inhibiting the inhibitors of the cyclin kinases. What's good about this microRNA is that about 70% of the entire repertoire of embryonic stem cells is by these microRNAs. They don't express many microRNAs, and 70% of all microRNAs in these exosomes belong to 290 to 94 family. When we transplanted this ES cell in the myocardium, and we look at the myocardium on day 5 post-MI, we can find the evidence of 294 expression and 291 expression in the myocardium. Now, these microRNAs are not expressed in the adult. They only express in the embryonic life. So it's a very neat system to really show that these microRNAs in the myocardium are coming entirely to the transfer by the ES exosome compared to the original microRNAs. Then we, of course, begin to look from the exosomes to just this microRNA, one microRNA from this family, to show that at least some of the effect of the ES exosomes are mediated by this particular microRNA. This is shown here is that mimic treatment of microRNA, or where we're looking at the cell cycle of CPCs. As you can see, when you treat the cells with this microRNA, they have a much shorter G0 to G1 cell cycle phase with a larger S phase. And also, if you look at the changes in the cell cycle genes, microRNA, both 291 to 94 treatment enhances the expression of the cyclins into the cells that were treated with the mimics of 294. Treatment with 294 also enhances reduces the cell death, improves the survival, as well as the activation of number of cell survival activating cell survival proteins, including phospho-AKT and the Lin28, which is the direct target of this microRNA. We also created a stable cell lines where we overexpressed the microRNA 294 in a continuous fashion, and these lines show much better proliferation activities, as well as much smaller population doubling time and much better metabolic oxygen consumption rate. So this also looks like this particular microRNA changes the functional capacity of the cells very well. So what I've told right now is that the adult heart, the CPCs are affected by the age and disease. There's decreased survival. This microRNA 294 through its exosomes leads to a transient decapitulation of the embryonic or developmental signaling, including the expression of Lin28 and the target genes, which leads to the enhanced CPC proliferation as well as myocyte proliferation, enhanced potency of the CKS exosomes, and altogether this process may restore the endogenous cardiac repairability. So this is all that we published. Then we begin to look at the microRNA in mode of a temporal expression. This is just to show that this microRNA is very highly expressed in embryonic life, but as just after day, this expression goes down, and by day 21 postnatal life, you can hardly find this microRNA's expression in the heart. This is also to show as this microRNA goes down, so does the expression of the Lin28, which is indirectly activated by this microRNA. And we also show that same thing with the mimic treatment. That when you put the mimic in the neonatal cardiomyocytes, you can increase the expression of Lin28, as well as in adult cardiomyocyte in terms of RNA. When you put the mimic, you can see this, the expression of Lin28 goes up. We also did some functional assay with microRNAs in cardiomyocytes. This is the neonatal cardiomyocytes where the treatment with the mimic enhanced both the Ki67 positive proliferation of the cells as well as the cytokinesis shown in here by the expression of Aurora B in myocytes, and in addition to the expression of the cell cycle genes in the cells that got this mimic of this microRNA. Not only in the neonatal cardiomyocytes, we also use the cat, feline cardiomyocyte adult, and if you treat them with this mimic, you can also activate the phosphorhistone 3 staining, suggesting that these cells are going into the cell cycle, as well as the cell cycle expression genes. Then we directly tested this microRNA, and this is ongoing work, and we have not published this yet. This is a single, LNA-based IV injection of this microRNA, and even at one dose, you can see that it's trending to be much more functional, or at least mimics some of the effect of the ES exosome transplantation myocardium, both in terms of function and structure, as well as reducing the cell death and augmenting the proliferation of the myocytes and CPCs in the heart. So it appears that ES exosomes largely function by this family of microRNAs that are exclusively expressed into the developmental stage, and they probably recapitulate the embryonic signaling into the adult heart. So that's what is shown in here. So so far so good. Now in next 10 minutes or so, I will try to finish about this cautionary tale. The question we begin to ask was this: are all stem cell exosomes created equal? And we already know, and the thought came from, it's all unpublished data, so physiological stresses like systemic inflammation and diabetes, they establish suppressors of the stem cell biology and function. So what we hypothesize, and we use bone marrow progenitor EPCs or endothelial progenitor cells as a model system for these studies, just because we have the animal models that can use. And we hypothesize that these EPCs under stress of inflammation and diabetes may modulate both the content and the function of their exosomes. This is just to show some of the cell data we published long time back, and this is, we use the interleukin-10 knockout mouse as a model of systemic inflammation. These mice are highly inflamed, and they develop cardiovascular as well as other autoimmune diseases. And we took the EPCs from these ALT knockout animals, and these EPCs are not angiogenic at all. They do not form any tubes in vitro. And if you take these EPCs and you inject into the heart, they are dysfunctional. They don't improve any function compared to wild-type animals, as well as they are less angiogenic, shown in here in terms of capillary density in the border zone, as well as the infarct size. So these cells are dysfunctional. We use these cells to see whether the exosomes mimic what they show in terms of their function. We also use the diabetic DB EPCs, and these are even worse than the IL-10. These EPCs never try to form the tubes by themselves. They have defect in the migration as well as function. So this is as the diabetic EPCs put back into the diabetic animal in the hind limb ischemia model, as shown here in the laser Doppler imaging, that these DB EPCs are dysfunctional compared to the wild type counterpart. And this is just shown the capillary density. So we already had evidence that stem cells coming from the inflammatory milieu or the hyperglycemic and diabetic milieu are dysfunctional. We wanted to show whether the exosomes from these stem cells are going to mimic or phenocopy their parents' functional ability or not. So the way we define EPCs is this is the bone marrow population, which is positive for the SCA1 as well as C31. So that's the endothelial entity and stem cell. They're about 4% population in the bone marrow, and that's what we call them EPCs. They exhibit all the characteristics of EPCs. We are exosome from them. They are all exosomes. We characterize them by both protein analysis as well as the facts. They express CD63 and CD81, which are the exosomal markers. We also did the particle concentration by nanocyte. They are about very highly expressed in these cells, and then they have about 16 nanometer size by DLS analysis. When these exosomes are very readily taken up by the recipient cells, they deliver both protein and RNA. There's a case that you can label the content. The red is in the RNA and the green is the protein. And if you look at the cells, they both fluorescence is there. So these exosomes have both proteins and RNAs. And this is just the protein concentration per particle number size. So there's a linear relationship. In the functional assays, if you do a cell death assay in of the endothelial cells and you treat them either with the wild type EPCs or with the IL-10 null EPCs, you can see that not only they don't protect like the wild type, but they actually enhance the cell death if you put the exosome from these stem cells. Similarly, in the tube formation assay, if you do HUVEC, you put these EPCs on the HUVEC, they also tend to disrupt the tube formation. So rather than not being functional, they are disruptive. And this is just to show the same thing that happens with the DBDB EPC-derived exosomes in the enginetic model system. We also use these exosomes for the myocardial infarction model. This is just to show that they're taken up by the host myocardium, and as you can see, the EPCs compared to the wild type stem cell derived EPCs, the IL-10 null EPCs as well as DBDB exosomes are completely functional. They do not mount any protective response in the myocardium. And it's the histology here. I think I'm running a little late, so I'll just skip some of the data here and just show you that if you look at the content of these exosomes, this is just the IL-10 knockout exosome versus wild type. They're very drastically different in the loading of the content. They're same stem cells, only coming from a different milieu or different microenvironment. And you see this very drastic changes in the microRNA contents as well as protein contents. This is a very crude Kumasi stain of the exosomes. And you can see they have a number of proteins that are now de novo expressed in these EPCs. So what I'm trying to say is that the content and the function of these exosomes dependent upon the microenvironment where the cells are coming, and they most often phenocopy the functional deficiency that is in their cell. We look at the proteomics too, and just to show that they have a differential amount of enrichment of certain proteins, and we are working on the functional component of these proteins in here. They also changed the signaling component. So this is an experiment where we use the EPCs from this null mouse exosomes and put them on the macrophages which are not activated. You can see the exosomes alone can activate the nuclear translocation of the NF-κB pathway and the activation of the NF-κB dependent gene. So in the absence of the ligand itself, just the content of the exosomes is sufficient to activate the signaling pathways that is activated in the parent cells. Similarly, you can modify the content. So one of the microRNAs is microRNA T75 is very highly expressed in the IL-10 knockout cells. If you take the antagomer to the cell and now collect the exosomes, you can drastically reduce the expression of this microRNA, and that way you enhance the function of this. You can rescue the phenotype of dysfunctional exosome by either modifying the cell themselves before you collect the exosomes. Not only in vitro but also in vivo. If you inject the now modified exosomes that do not have microRNA T75, you can rescue their function both in vitro and in vivo. This is all a very preliminary experiment that I'm showing you. This is just to show. I still have 5 minutes or so. 5 minutes. All right. And this just show that this augmentation of the downstream signaling pathway when you inhibit this microRNA, you enhance protective signaling both in vitro and in vivo. And similarly for the diabetes, they also, if you take the cells, both the human and the mouse, which are under hyperglycemia from DBDB mouse, You see the enrichment of certain microRNAs, which are known to be elevated in diabetes. And you can modify, you can change. You can either now, you can also use the integument to directly transfect the exosomes and rescue their phenotype shown in here. You don't have to go to cell. If you identify certain microRNAs that are different in your exosomes, you can directly transfect the exosomes loading by the integument or mimic as you wish. And when you apply them on the cell biology experiments or in vivo, you can rescue the defective phenotype. The same is true for the protein expression as well as this epigenetic expression. It shows the repressive epigenetic marker, both the NMT3 alpha as well as TAGs, are highly enriched in diabetic cells, exosomes. When you put on the cell, you can see that the endogenetic genes have shown very reduced activation in the chip of histone 3K9 asparase, which is a positive mark for the gene expression. So what in the second part I told you is that we have to be cautious before we begin to think about applying the exosomes in the cell therapy or replacement of cell therapy. Clinically, exosomes, even from the same stem cells, they are not created equal. And cells phenotype and environment dictates both their contents and function. And I'll just end up here by acknowledging my collaborators as well as my funding agencies. Thank you for your attention. Yes, Dan. Right, so I'll take your second question first. They are mildly immunogenic. The way they are made is by the invagination of the plasma membrane, so they do carry certain load of the MSC, class 1 and also class 2 to a certain extent. But the total load of MSC on the exosomes is about 1/100th of the cell itself. So they are mildly immunogenic, but they haven't been, in our experience when we use the human exosomes in both mouse and pig, we did not find the augmentation of this T cell or B cell or normal responses. They might be immunogenic, but no more than the MSCs, I guess. And the CPC question is this: the reason we primed CPCs was because we saw the great number in augmentation both in the number and the proliferation of CPCs. When you put CPC exosomes, EC exosomes, you basically are reprogramming these CPCs to be the super CPCs. So the functional change you're seeing is probably not just by the cells themselves, but now the CPCs are also exporting the exosomes that have a component of the ES cells. So that's probably the hypothesis. We haven't proved it yet. So they have the similar structure in the plasma membrane, just like a bilipid layer, just the cells have. There are regions similarly you transfect your gene of interest. The genes, the big DNA, they might not carry because of their size, but the small peptides and the microRNA specifically, the integument, they can directly mix the regions together in test tube. They very efficiently. We have done like a lot of fluorescent microRNAs. They all just go in very quickly. So that's, we can directly do that. Pardon? Yeah. When you stop the embryonic stem cells before you collect this, so that's a good question. There's some evidence that both the functional capacity of other adult stem cells is improved if you take the exosomes when the cells are in the starved condition or cells are in the hypoxic condition. But with the ES cell, we haven't collected the exosome when the ES cell is starved. So we don't have the answer to that data. Half-life in terms of what? So they are very, very stable. So they don't pop. Once they are fused to the membrane and they go into the cell and digestible lysosomes and deal with the content, then as soon as cells take them up, probably are digested and deliver the content. In vitro or ex vivo, if you take, they're very, very stable. It's much more stable than the cells. You can just freeze and thaw them multiple times without rupturing their bilipid membrane. No, not yet. But I can just, what we have some data. I'm telling you the bottom line is that gender does matter.
Stanford University.